Hello, I'm Harold from Promega. Today we are in Professor Oliver Mühlemann lab at the University of Bern, and we will speak with team members how they will use the Maxwell in their daily work. Okay, let's go. Hello Nicole, how are you?
Hi Harald, nice to have you here. I'm fine, thank you.
Can you tell me a little bit about your lab?
Our lab is investigating post-transcriptional gene regulation in mammalian cells. So a main focus is on trying to understand nonsense-mediated mRNA decay, and this is a mRNA quality control pathway that recognizes and eliminates mRNAs that fail to terminate translation properly. So kind of we are a typical RNA lab.
So how did you hear about Maxwell? I'm just curious.
Because we are really doing a lot of RNA extraction. For many years we did classical RNA extraction, so specifically tri-reagent extraction. And our people are really good at this technique. But I thought, well, each time they have, for example, 16 preps, it's about 2 hours, 2.5 hours hands-on, you know? And this is a lot of workload, and specifically the postdocs, you know, they have a lot of tasks. And I just thought, maybe we could do something to make their life a bit easier. And this was how I started looking around the market, what robots are available that could do RNA extraction for us. Yeah, only the Maxwell performed really well, you know. No, it works just well, and our people love it. I mean, they load it with RNA, they go away for 1 hour, you know, and this 1 hour allows them to do whatever else, you know. They can read a paper, they can write their lab journal, or focus on anything else, but don't have 2 hours hands-on, you know. And so this frees up the people, and yeah, it's a relief for postdocs and PhD students. Yeah.
But in a nutshell, you're happy with the Maxwell?
Yeah, super happy. Super happy. Because the quality of the RNA is consistently super good. Super good. And also, you know, we had a special application where people wanted to purify minimal amounts of RNA from in vitro reactions. And indeed, this only worked with the Maxwell. This did not work with the tri-reagent protocol.
So we should look at this workhorse Maxwell in the lab?
Yes, sure.
To watch user Evelin, how she
go.
manipulates with the Maxwell?
Let's go to the lab.
Hello Evelin, nice to meet you.
Hello Harald, thank you.
How do you use the Maxwell in your daily work?
So I use the Maxwell for RNA extraction, and the RNA I then use for RT-qPCR later on.
And do you isolate the RNA from which kind of samples?
I isolate RNA from hex cells or mutant cell lines which have parental hex cells, mainly, yeah.
And can you tell me what you like especially about this Maxwell instrument?
So I like that it's very easy and it's very quick. So within an hour itself, the RNA is extracted, and before that it's like 2 or 3 steps, and those steps are very easy, and there's no toxic reagents that are used, which makes it very friendly to use.
Thank you very much for the interview.
Thank you very much for coming.
Yeah. And I wish you all the best for your future.
Thank you very much.
Thank you for joining us. I hope you learned something about Maxwell. See you next time.